So I think we can get started.
Great.
Okay. So good afternoon, everyone, and then welcome to the Statistical and AI Methods for Health Data Science Seminar Series. So the goal of this seminar series is to introduce various kinds of biomedical data to the statistical community, and then discuss current advances and challenges in the fields of statistics and AI, and then also discuss the future directions. Just a brief introduction about the StatsUp AI group. So it aims to organize activities that empower statisticians to participate more actively in AI research and then leadership. And then just a good news that this group has been officially recognized as an ASA interest group. Now here is the website, and then you can also sign up the Google group for StatsUp's AI StatsUp for AI. And then here is also a brief introduction about the steering committee and then the inaugural officers. Today, it is my great pleasure to introduce our speaker, Dr. Ying Guo. Ying is a professor of biostatistics and bioinformatics at Emory. She also has a joint appointment as a graduate faculty of the Emory Neuroscience program and an associate faculty of computer science. She is a founding member and then currently serves as the director of the Center for Biomedical Imaging Statistics at Emory. Ying is widely recognized for her pioneer research in blind source separation methods, especially in the development and application of the ICA-based methods for fMRI data analysis. Recently, she also made significant contributions to the field of longitudinal imaging data analysis and multimodal data integration. Ying also has made great contributions to the imaging community through both her scholarship and then leadership. She's served as the 2023 chair of the ESA Statistics and Imaging section, and then also organized the 2021 Statistics and Methods Imaging Conference, which is the annual conference of the section during COVID period. And then in addition, Ying also contributes to the broader scientific community through editorial service in both statistics and psychiatry journals. She currently serves as a standing member of the NIH EITN study section. Just before handing to Ying, and then maybe you'll miss some logistics of the seminar today. So if you have any question for her, for Ying, please type in the chat, and then we will save all the questions to the end of the seminar. And then at the end of the seminar, if we have time, we probably also have the chance to have you to ask the question. So then please raise your hand, and then I will call your name and then ask the question. And then I will stop sharing and then hand it over to Ying. Ying, it's all yours.
Okay, thank you, Yi. Let me share my screen. So am I sharing my screen, all right?
Yes.
Okay. Continue to share. All right. Well, thank you, Yi, for the great introduction. And I'd like to thank, first of all, thank you, Hongtu, for organizing, leading this initiative of Stats AI. And I think it's a very important initiative and very timely, for the current era. And I'd like to thank Yi, Edgar, and Pan for the great organization. You guys have been doing great. And I saw the list of upcoming speakers and I'm really looking forward to the following talks. So today I'm gonna mainly focusing on, exploring brain function, especially connectivity using fMRI. And I'm cover the topics from data characteristics to the analysis downstream. Okay, so our interest here is to study the human brain, which is a very complex organ, the most complex organ in our body, and controls all our body's mind and activities from very primitive, motor functions to high order cognitive, functions. So when you're looking at the brain, it's a 3-pound of fatty tissue, and most of the neurons are located on the cortex, which is the outer surface of the brain, which is as thick as 4 sheets of paper. So yeah, if you kind of like open up all the, the cortex regions and then smooth out all the foldings, the area of the cortex surface is about the same size of a baby blanket, basically. So Okay, so before the fMRI, people have already been very intrigued by the brain functions. So here are some early understanding of brain activities and localized brain functions. And the information back then was mainly derived from, some lesion studies by looking at people who had a stroke and see, what part kind of function was affected, or through some kind of postmortem, brain studies. And with the development, actually, advances of neuroimaging, especially fMRI back in the 90s, the understanding of a human brain has advanced significantly. Now we have some more detailed and precise understanding of various of brain functions in different brain locations. And more importantly, through the fMRI data analysis, we now derived this very new knowledge about how different brain regions connected and communicated with each other and synchronize the activations and divide functions in different networks. So let's first take a very brief look at, the brain construction. And there are two types of brain tissues, gray matter and white matter. To understand them, let's go back first to neurons, which is like, the cells in charge of our neural activities in the brain. Here's a picture of a single neuron. It's mainly composed of 4 parts. The dendrites basically is this tree-like part of a neuron. They basically are responsible for receiving signals from upstream neurons. After they receive those signals, they pass that information to the second part, which is the cell body. This is where the nucleus of a neuron is, located. And here the cell body will take the signal and do some processing and decide whether it will take any action or not. When it decides to take action, then it will generate action potential, which are electronic pulses. And those electronic pulses are passed down or transmitted through the third part, which is this long projection called axon. So the axon basically is a fiber that can transmit those electronic signals and is wrapped by this myelin, which is a kind of white fatty tissue to help it to increase the neural signal transmission speed. Then the fourth part is axon terminal. Once it receives the signal, it will remit neurotransmitters, basically allow it to convey information to downstream neurons. So when you're looking at the brain, like the dendrite cell body and axon terminal, they basically consist of the gray part in the brain, which is we call the gray matter, that is most constitute outer surface of the brain. And also we have some subcortical gray matter as well. And then underneath the cortex in the brain, you can see those white region, and that's what we call white matter. So the white matter essentially are formed by the bundles of those axons. Since the myelin, the white fat tissue wrapping it has the white color, that is how we get this white matter, composition underneath the cortex. So we can view the gray matter as kind of like a CPU in a computer if we view the whole brain as a computer, basically. The gray matter is kind of like the CPU where the neural activity took place. And the white matter is kind of like the cables in CPU where the information was transmitted from one brain region to another brain region. So we use different imaging modalities to study the different tissues, like for the gray matter where neural activity happens, fMRI and other functional imaging is used. Well, for the white matter, diffusion MRI is used to study those white matter fiber connections. Okay, and when we look at fMRI, when people take the fMRI images, there are typically 2 different types of MRI images acquired. First, we will obtain this kind of a structural MRI image, followed by a series of functional MRI image. So what happened is that we change like the sequence in the MRI machine, so to capture different types of contrast. So the structural MRI captures the T1 contrast, so they form this T1 image. The T1 contrast, basically the characteristics is that you see bigger difference in the signals between different brain tissues, like gray matter and white matter are well separated in a T1 image. The downside is take a long time to acquire a single T1 image. The the upside is provide some high spatial resolution that distinguishing different type of tissue. So we usually obtain one structure image, T1 image in the session, and this is what we call the structure image because it helps to study the brain's structure. And then the series of functional MRI images are acquired when we change the sequencing to capture the T2 contrast in the MI signal. So the T2 images are obtained much faster, per image. And the downside of it is that each one of them has a low spatial resolution. The difference in the signal between different brain tissue is much smaller. So that's why the different tissues are harder to be distinguished from each other in T2 images. But since it can be captured very repeatedly, like say every 2 seconds, you get a whole brain T2 scan, we use it to kind of do the functional MRI scan of the brain. So the fMRI basically is the T2 weighted brain images, MRI images. So basically, it has become a very prominent, one of the most commonly used imaging modalities to study brain function and activity. One of the main strengths of MRI is that it provides a very attractive balance between spatial and temporal resolutions. So it offers a very good temporal resolutions that's good enough for us to distinguish the brain activities under different types of tasks or under different kind of cognitive states. But at the same time, it also offers a spatial resolution that's good enough for us to identify the spatial location where those neural signals originated from. So because of this good balance, attractive balance, fMRI has become a very powerful tool. So basically, when we're looking at the mechanism underlying fMRI, it is an indirect measure of brain activity. So unlike EEG or MAG that measures the neural electronical, action potentials generated by the neurons directly, fMRI actually measures a correlated neural activity, which is the hemodynamic response. What happens is like neurons, like all the cells in our body, when it functions and gets active, it consumes more energy. Then the body would respond by sending like more oxygen-rich blood to the local region. So this will lead to an increase of oxygen hemoglobin in the local area where the brain activation is taking place. So it turns out that here's basically like the normal blood flow where the brain is not activated. So you see the kind of like the blue dots, and the red dots, where the red dots represent oxygenated hemoglobin, while blue dots represent deoxygenated hemoglobin. So these two types of cell they actually have a different magnetic properties. When the brain gets activated in this region, you would see more oxygenated hemoglobin in the blood. That will lead to an increase in the MI signal. So it is because of this change related to the blood oxygen level that lead to the change of fMRI signals that correlate or reflect the underlying neural activity. Here's a typical pipeline for fMRI analysis. So we start with imaging collections, and we have different type of study designs, and the acquisition will be different. And basically, after that, we'll do imaging processing. And before the analysis, we actually need to do pre-processing and some quality controls of image, which is a very crucial step and have very strong effect on the downstream analysis. And once the image are properly processed, then we'll take it to some analytical to be analyzed through some analytical techniques, including statistical methods, as well as AI methodologies, in order to achieve specific tasks. So for fMRI analysis, the three most commonly conducted tasks include study the local activation and see that's okay. Study the local activation and sorry. Okay. To study the local activation, basically to study what brain region is activated in response to a specific task. The second type is study connectivity between different brain regions, local regions. The third type is to using some fMRI extracted features or images to do some downstream predictions, for example, to predict subpopulation groups and to predict some clinical outcomes. So I start with the study design of fMRI studies. There are typically two types of study designs, that is a task fMRI and resting state fMRI. Task fMRI was the first type of study design that came into place when fMRI was first used, and we started with block design specifically. So the task fMRI is that you would give the subject some specific task when they are being scanned in the machine, and they're trying to study task-induced brain activity. And the main purpose is to identify what brain region is involved in some specific cognitive or motor tasks. When we look at how the tasks or stimuli are arranged, during the experiment paradigm, there are different type of designs, including block design. Here I have a picture of it, where you group the similar types of tasks together into the same block, and then followed by the resting or condition or control period, control block. So here, this is a face matching task. During the task block, basically the subject will see, a series of presentations in the computer screens where the faces are presented. And after this task period, they're followed by the control period, control block, where they will see some, control visual stimuli without faces. So basically, this is what we call the block design because we put the same type of stimuli into blocks. The advantage is that you're going to see some, stronger signal between different conditions. And later people have invented event-related design. So that is those different tasks and control conditions, they are not blocked. Rather, they'll be interleaving with each other. So one of the challenges of fMRI data is the no signal to noise ratio. If you're looking at the fMRI signals between an active task and a control condition, the difference is very small relative to the overall, variation in the fMRI signal. So for block design, the signal to the SNR is about 3 to 5%. For event-related design, that often is less than 1%. So that is why it is so challenging to analyze fMRI data. And for task fMRI, the main study analysis goal is to study the activation, like a GLN type of regression and other type of statistical inference has been used. The main feature that has been extracted from task fMRI is this kind of contrast maps, where you basically comparing the images under the control condition versus the task condition to get this contrast or T-map, we often says, that is used to, kind of predict some clinical outcomes or group memberships. Since the task fMRI, people accidentally discovered actually, even when subjects are not doing those active tasks, there are some spontaneous brain activity taking place in the brain. Specifically, they found out like the left and right side hemisphere, similar functional regions in the brain, they would have highly correlated fMRI both signals under the resting state condition. So those reflect there are some intrinsic spontaneous brain activity taking place there. So the resting state fMRI since then has been used to study those intrinsic net organization of the brain. The main purpose in resting state fMRI, since there's no task, is really focused on studying how the different brain regions are connected. So connectivity study is really a big thing for resting state and the main type of analysis for resting state fMRI. And there are some brain activity measures that you can use to measure basically the activity during the resting state. But the most commonly used features from resting state fMRI is the resting state networks. People through analysis, like I say, have identified some very reliable set of functional networks. They are large scale and spatially distributed across the brain. And then they basically perform similar types of brain functions. So the most famous one is the default mode network, for example. Okay, so when we're looking at fMRI data structure, they look like this. It's basically a 4D data. It's a time series of 3D brain images. At each time point, you acquire this 3D brain images across the whole brain, and then you do it repeatedly across time, basically. So that's why we often say fMRI data is 4D. Each brain image can contain hundreds of thousands of voxels, and the number of scans you capture over time in one session often can be in 200 to 400, for example, typically. Okay, and when you condition or specific, focus on specific locations in the brain, you're gonna see this time series of both signals representing the neural activity in this location. And for this 4D type of data, people often represent it as a 2D matrix by flatten out the 3D matrix obtained at each time point into a vector. So we typically form this a T by V matrix, where T is a number of scans you obtain over time. V is a number of voxels you obtained at each time point. So therefore, at each row, you have this time series at a specific brain location, and each column, you have this 3D map, basically, that is acquired at a specific time. So in fMRI data, we have complex correlation structures. So we have the temporal correlation. So over time, we also, given a specific time point, you have the spatial correlation structures. So that's why the fMRI data is said to have complex spatial temporal correlation. And that's what's making its analysis challenging. Okay, so next I want to talk about the imaging processing of fMRI data. Okay. As I mentioned, one of the challenge in fMRI is that it's indirect measure of neural activity. So there are a lot of sources of non-neural related variations. So the brain itself is very noisy, and the signal of interest is weak, and the scanner will generate some noise. Basically, as time goes by, for example, the scanner may get hotter and hotter. That will give rise to some kind of low frequency shift in the intensity in the fMRI time series. And also the subjects have some physiological variations when they take fMRI images, like our heartbeat and breathing. They kind of will generate some variations in fMRI signals in the and also the movement is a big source of noise in the fMRI data. So the solution to that is that in addition to try to ask the subjects not to move, as best as they can in a scanner, pre-processing is a very important and crucial step that help us to systematically remove those non-neural variations and kind of like clean our data to facilitate downstream analysis. So during the fMRI scanning, the subject typically will first take this T1 image, which is the structural image, followed by the series of functional fMRI image. So the pre-processing is performed on both the T1 image as well as the subsequent fMRI image. And those steps are kind of like interactive between the two types of imaging modalities to register, different imaging modalities. Okay. So here are the typical steps of fMRI pre-processing. We correct the slice timing difference, and we collect the motion effects and register the function and the anatomical data. And we also spatial normalize the scans because everyone's brain is different. So we normalize them to a standard template, facilitate group analysis, for example. Spatial smoothing is done mainly to increase the signal to noise ratio, basically, but it does have some consequences for the subsequent analysis, especially connectivity analysis. And following those standard pre-processing, depending on what kind of fMRI you are doing, then we have some specific pre-processing follow-ups. Like for task fMRI, we would convolve the stimuli with the HRF or hemodynamic response function, and to generate what is the expected fMRI signal after a neuron gets activated. And that will be used for the regressors in the subsequent GLM analysis. For resting state of fMRI, we need to conduct some additional steps to further clean up the data. So one of the important things is the nuisance signal removal, which has a very big effect on the fMRI data. It's a very crucial step, for resting state fMRI. Basically, is that we will take those signals in the white matter and fluid CSF regions where there's supposed to be no neural activity, take place, and therefore those signals kind of act like a nuisance biomarkers. They reflect what are the nuisance signals in the brain. And then we will take that and remove them through regression. And temporal filtering, which is a band filtering, because most of the resting state of fMRI activities happen in a specific bandwidth. So by doing the band filtering, we remove those high frequency noises. And another important step that is done is motion effect control, because motion is a big factor that, deteriorate fMRI scans qualities. So there are various methods proposed, like confound regression with motion parameters to regress out those motion effects. And temporal censoring basically is to identify those scans that is contaminated by motion, and then you either remove them or interpolate them. So some of the commonly used methods are here. And later on, people have proposed to use ICA type of method to identify those components that is related to motion and remove it from the data systematically. So the current motion effects control method, they are very intuitive and works very well, but they do have limitations. For example, those temporal censoring would lead to a loss of a lot of scans, in the time series, and also the loss of many subjects, where we have a certain threshold of motion control. So to address those limitations, I have seen some advanced motion control methods that are being developed by the statistical communities, and including some methods developed by my colleague Berisk at Emory, where he uses causal inference methods as a way to address emotion. And then I heard a recent talk by Zhenwu Zhang that uses deep learning to try to kind of generate fMRI images and features where there's no motion. And so those kind of advanced method I think, can potentially offers much more better, improved solutions to this very challenging problem. So here I just want to use some kind of results to highlight why the pre-processing is very important in fMRI analysis, and it's something we should pay attention to. And that is because they have some very profound effect on the imaging fMRI features we extracted. And so here we have a looking at functional connectivity matrices that is estimated from a subject using the same subject's data. On the left one, we estimate the FC without doing a global signal regression. So global signal regression is a pre-processing where you remove the overall bold signals across all the voxels in the brain. So you calculate the time series of overall, signals across the whole brain, and then you would use regression to remove that global signals from your fMRI data. So by doing so, we basically help to remove some spontaneous bold signal fluctuation common to the whole brain, which is believed to be not necessarily, neural related. And so basically, the advantage of it is can help us to increase the specificity of the FC analysis and help us identify local, neural activities. Without doing this GSR, you can see the FC matrix is all very positive all over the board. So that is most of the connections estimated are positive. You see this whole red, FC matrices. That is because you didn't remove this global effect from all the brain regions. After you remove this common global confound, you can see the picture is much more balanced between positive and negative associations. So one of the downside of GSR is that you can show statistically when you do this global signal regression, you would actually you would mandate some negative functional connectivity in some parts of the brain for some of the brain connections. So therefore, it is criticized for inducing some negative functional connectivities. Okay, so it's a double-edged tool. However, most of the imaging practitioners, they believe that not doing a GSR, you have a bigger harm because you don't remove that global signal and global confound. Okay, so but when comparing these two different FC matrices, you can see how much these pre-processing steps effect have on the our measures of connectivity. So here's another example which shows how important to have the same pre-processing across different studies. On the left, we have this is a group level FC functional connectivity matrix that we estimated using the human connectome development study data. So, and on the right is our FC matrix that we estimated from the PNC study, which is another development study. So initially, when we're looking at these two matrices, we see, oh my God, they're so different, right? So what are that seems to suggest there are some major differences between subjects in these two studies. However, then I realized actually our PNC data was pre-processed using an early pipeline. So after we change it to using the same pipeline as in the first study, then this is what happens. And one thing we have noticed is that now the connectivity matrix in PNC studies is much more comparable to the other studies. So therefore, in a multi-study analysis, no matter what your goal is to compare results between two different studies, or your goal is to combine the data from different studies, combine the functional connectivity from different studies, is really crucial for us to implement the same pre-processing pipelines, because otherwise you would see some differences that will derive from the different pre-processing that will confound your subsequent conclusion. Okay, so after realizing the huge impact of pre-processing on the downstream analysis, the neuroimaging community have converged on using standardized pre-processing pipelines. And so I remember like probably 10 years ago, or when I first started with fMRI, when we read the paper of fMRI paper, the analysis of the pre-process steps is often done in very different way, quite different way from one paper to another paper. And also each lab, they have their own in-house pipelines in terms of how they're pre-processing. And in recent years, we have seen like there's this method, for example, called fMRI prep, which is a pipeline for pre-processing fMRI data. And it has become the dominant method for pre-processing fMRI data. So nowadays, when you read a current paper, most fMRI pre-processing are done through this pipeline. So this really helps to make the results from different fMRI study to be more comparable to each other. Okay, and the same lab also has released a subsequent pipeline, which is to do the quality control for fMRI scans, basically. And what happens is that when you do the quality control of fMRI, structural MRI, many of it involves some very tedious visual recognition or visual inspection of scans across individual subjects. So what this pipeline did is that, first of all, they automated those visual inspections. And so the results are very systematically generated and provided. So you don't have to do a lot of, time-consuming clicking box to see those results. And secondly, they have automatically generated some imaging quality control metrics. So, and then they output those metrics so you can get some very quick idea about which subjects has, too much motion and needs to be, reconsidered, and what is the overall, quality across different, different studies, etc. So this pipeline has been very helpful, in terms of doing the quality control of the studies. So that is the second part of in the fMRI analysis procedure. And thirdly is after we have pre-processed those fMRI images, and now they are clean and they are in a good, in good shape, then we can take it to do the subsequent analysis then. So as I mentioned, there's 3 different types of analysis typically done due to the time constraint here. I will focusing on the connectivity analysis using fMRI. Okay. So the brain connectomics, or the brain connectivity analysis, or brain network analysis, often they refer to the same thing. It has attracted a lot of attention in the current neuroimaging community. Is one of the most commonly done, analysis nowadays in fMRI. And so basically, this is a field that is focused on comprehensively mapping the interconnection across different brain regions. Okay, and so we can, depending on what imaging modality you use, you can generate functional connectome or structural connectome. And then once you generate that, we use those connectomes for some, different type of analysis to understand cognition behavior, to study neurodevelopment, aging, and to identify brain markers for, disorders, and improve our personalized diagnosis and treatment predictions. Okay. Okay, so depending on what type of connectivity that we estimate, so there's the structural connectivity that is measured using diffusion MRI, and the functional connectivity and effective connectivity that is measured using fMRI. So the difference between functional and effective is whether the connectivity is undirected or it is directed. Okay, so like for the fMRI, basically our analysis is based on fMRI time series, and from that we generate FC matrices. So in the brain network, basically each nodes represent specific regions in the brain, and each edge in the network represent the connection between a pair of nodes in the brain. So for functional networks, we generate this from the fMRI data, and for diffusion data, we generate what we call structural connectivity matrices, which reflect those white matter fiber connections between different brain regions. So here in today's talk, I'll focus on the functional connectome analysis. So for this type of analysis is mostly composed of two steps. First step is to construct the network. We start from the fMRI images. We typically apply some kind of brain parcellation or atlas, that divide all the voxels into groups of voxels, which we call the regions of interest or node or puzzles. And then for each of the region of interest or node, we extract the representative fMRI baud series. And using some kind of dependence measures, we measure the pairwise connections to generate these connectivity matrices. And through some thresholding, we will lead to a brain network. And once you estimate the connectivity and network, we typically take it to do some kind of a statistical and AI analysis to study brain disorders and other type of outcomes like neurodevelopment and aging, etc. So let's look at the network construction. As I mentioned, the first step is to implement some brain atlas or parcellation. There are different types of brain atlas that's been currently used in FC analysis. The first type of analysis people used is AAL, those kind of anatomical parcellation. And when you look at the earlier FC papers, most of it is based on AAL parcellations. So in those anatomical regions, those regions are defined based on anatomical information. However, each of those regions in anatomical atlas, they do not necessarily share similar functionalities. So that is the downside of atlas, using anatomical atlas for functional connectivity analysis. After realizing that limitation, people have generated quite a series of functional based atlases. And so in these atlases, basically they are generated using fMRI data, a lot of it using resting state fMRI data because that reflects their intrinsic brain connectivity. And then they partition those brains into those functional regions, putative functional regions or puzzles based on the functionality. So voxels in the same puzzles, basically they are having usually have a similar type of connectivity profile with the rest of the brain. So that's how those regions are defined. So because those regions are defined using functional data, they have a better alignment with FC analysis downline. And in addition to use those pre-specified atlas, there are many studies that actually use a study-specific atlas that is generated by applying ICL on the current study. Well, the motivation is that in some of the studies, the population is quite different from the populations that were used to generate those public atlases. So which would affect, the accuracy of those functional areas in capturing the neural activity in that specific study. So therefore, in some study where the population is very specific or different, they actually would do, their on-site parcellation. Okay, so here there are some papers that have studied the effect of choosing different atlases and how they affect, the downstream analysis and the functional connectivity derived from it. Sorry. So basically in this paper, they showed when you use different atlases, the average strength of the functional connectivity actually can change a lot. Sorry. Okay, from one atlas to another. So in another paper, they used the functional connectivity to predict what kind of cognitive state the subjects experienced. And they quad images under like a motor task, vision task, and rested state. And they use the FC data, functional connectivity data, to predict or to how accurately they can predict the specific cognitive states the subjects are experiencing. And they generate FC using different atlases and compare the classification accuracy. So one thing they observed is that if you're using AAL, those anatomical based atlas, the prediction accuracy of the FC matrices generated from such anatomical atlas is much lower than using those functionally derived atlases. And another finding they have shown is that using those ICA-derived, study-specific atlases, you can increase the prediction accuracy, of the FC matrices. So this basically shows the effect of using, appropriate atlas in a specific task. So, and in terms, once you decide, what atlas you're going to use, the next step is to determine what kind of measure that will be used to capture the functional connection and between different brain regions. So people have reviewed the whole, the spectrum of measures that is used to model the functional network modeling. And in this paper, for example, Smith and al. Have done some extensive simulation studies and evaluated different network modeling methods. And from the study, they found out those partial correlation-based methods tends to provide, better recovery of the functional connectivity patterns. Because comparing to full correlation, partial correlation remove out the other nodes confounding effect and potentially can better capture the true dependence between regions. Okay, and here I have a listed, this is an incomplete list of the different type of connectivity and network method that has been developed for us to study brain functional networks. And different methods, they have a different goals. Some of the method is trying to capture the whole brain connectivity matrices. Some of the method is trying to identify the brain networks by decomposing the overall data. And the graphic measures can basically measure modeling the network topology. And so depending on what your study purpose is, you can choose appropriate method for your purpose. So next, for the rest of the talk, I'm going to concentrate on the AI in neuroimaging. So in recent years, AI has caught a lot of attention in neuroimaging community and has, because it has shown great promises in other scientific field. So, and basically people have already found that AI can also generate some very exciting new findings for neuroimaging as well. So just to clarify on the different terminologies for AI, machine learning, deep learning, they are all used a lot in the field. So what do they mean exactly? And in this paper, they have reviewed the three concepts. So the AI actually is the biggest circle, represent essentially AI is an algorithm that try to mimic the human cognition and behavior or decision-making process. Well, machine learning is a subset of that use statistical method or computer science method to learn from the data. Well, deep learning is supposed to be another subset of the machine learning, which use complex computer science algorithms based on neural networks to train a model. So in this paper, they have surveyed the use of AI in neuroimaging analysis. We can see most often AI is used for classification purposes, mostly like classify disease groups, and for etc. And that is also used a lot in the lesion segmentation, like a brain tumor to study brain tumor, for example, and also for imaging reconstruction and regression and prediction. So for deep learning of neuroimaging has shown promising results in the diagnosis of different type of brain disorders. So in this paper, they have reviewed the use of AI deep learning method for neurological disease like Alzheimer and Parkinson disease, as well as mental disorders such as autism and schizophrenia. So in this table, they have summarized and reviewed the paper that use AI deep learning to study schizophrenia diagnosis, and it has shown some promising results in the classification for the schizophrenia patients, and especially when they are combining imaging modalities, such as combining structural MRI and fMRI. So we did some review of a deep learning method in brain network analysis. And here are some methods that commonly pops up in these areas. So basically like the feed-forward neural network or multi-layer perceptron, MLP, basically they are the early method that's being used in this area, and also with the autoencoder methods. And for those methods, often they take is a vector type of input. What they do is that they would vectorize, brain network data to go through the next step, deep learning structure. However, when you do that, you actually you remove those structure information in the network. So, and following that, like CNN, your convolutional neural network has been used to address that limitation, which will exploit, take into account of the structural information in the network. And graph convolution network has been used since then. And again, to take into account the graph structure, GAN has been mainly used for generative learning, mostly for data generation, data augmentation, and also generate type of synthetic image that can help them classify against control images, for example. And recently, there are some transformer-based methods that use attention mechanisms. So to learn the network information from a brain network, the advantage of the attention model is that they can learn the global attention mechanism. So in a sense that if you're using the connectivity matrix, you're only using a connection that captured by your existing connection, estimated from your data. But for the transformer model, what they can do advantage is that it can learn some non-local and complex hidden type of connections between different brain regions that may not be captured by the current existing connectivity measures. And so that is one of the advantage of transformer model. And also another advantage is it can learn some important connections that is important for downstream prediction tasks, basically. And the downside is that it's a very data hungry model. So basically reliably training them is still a big obstacle. In terms of the type of application in deep learning in neuroimaging, so here are the ones that we have found. Mostly it is for prediction or classification purposes. There's some paper doing the structural functional coupling, that is, they use a diffusion MRI data to try to predict, functional connectivity. So the idea is that the relationship between structural connection and functional connection are very complex. So it's basically, it's very kind of intriguing, because structural connection is supposed to drive functional connection, but the relationship is far more complicated than that. And to study this complex relationship, they use a deep learning model to predict FC from SC, for example. And it's also used in data augmentation and to increase the training sample size in the limited sample scenario. And another thing is used a lot is to do the imaging enhancement. For example, if to kind of like there's a deep learning method that is developed to generate a structural connectome from the diffusion MRI. So the goal is to overcome the limitation of the current, conventional tritography algorithm, so that we can generate more accurate connectome, structural connectome from the diffusion MRI data. Okay. So, and today I'm going to focus on two of the methods in the deep learning methodologies, because I think these two methods, they demonstrate to effectively use deep learning in brain imaging and brain network data. It is very crucial for us to develop some innovative strategy or adaptation of the deep learning architecture that specifically take into account of the unique characteristics of brain network. So here, the first example is a BrainNet CNN. BrainNet CNN was developed in 2017 and is one of the first deep learning methods that is specifically tailored for brain network data. And it has become very famous after its publication. And the main reason is that it uses some very meaningful neurological innovation to do the convolution for brain network data. So here's a classical CNN for standard images. So typically what they do is that they would use this kind of kernels, spatial kernels that take into account of the spatial localities to aggregate information from those neighboring pixels when they update the latent representation of the current pixel. Okay, so and convolve the information of the image. And however, for the brain network data, it's a different types of image. So brain network data is typically represented, as I just showed, as a squared matrix. In this one, they have a 90 by 90 brain connectivity data matrix. So you can view this 90 by 90 input as a typical 2D grid-like image. However, there are some fundamental differences. For example, the spatial proximity, characteristics that we observe in the typical 2D image, they do not transfer to the brain imaging data. In the brain imaging, the 2D grid, basically the spatial proximity doesn't mean the relationship. They do not code the relationship of those edges in that matrix. So in this one, in this 2D matrix, each element represents a specific pairwise connection in the brain, basically. So therefore, instead of use those special locality that classic CNN use, the BrainNet CNN actually leverages those topological locality between those brain edges in the convolution. So when you kind of like take each update element, each element which represent a connection between a pair of nodes in the brain. So when you define its neighboring elements, instead of looking just the elements that is spatially close to it in this matrix, we're looking at actually those edges that has a topological locality with the current edge. So those edges are those edges that shares one of the node with the current edge. So when you're looking at the connective matrix, that means the elements in the same row or the same column with the same current elements. So and those edges will be defined as the neighbors of the current party, and then their information would be aggregated or convolved to update the loadings, codings of the current, party. And then we do it again in another edge-to-edge layer in a structure. What you do is that you will take into the information from the second order, brain connection neighbors in updating the current edges. So you can see here, we use the topological locality in convolving the data across the brain network instead of a spatial locality. It has shown some great performance in predicting, the infant's age after using the DTI-based structural connectome data. And so it shows some promising results in some other applications afterwards. I think the reason why it's so successful is that this innovative topology, topological based convolution is making a lot of sense neurologically. Okay. So another emerging tool that is deployed to analyze brain network is a graph neural network. They have been used successfully to other types of network data. And basically one of the great advantage is that they can capture some complex high order network or graph structures in the graph. So however, the challenges for applying GNN to brain network data is that, first of all, brain network data to generate it, it takes a lot of complex imaging processing and construction, network construction, so which is a big hurdle for many computer scientists and statisticians. Secondly, the brain network has some different characteristics as comparing to other type of network data, such as social network. How to effectively design a GNN for brain network data, that's a big question because many of the existing model structure does not work well. And also the reproducibility of the analysis is a big question. So if we want to repeat one GNN analysis in another study, it's very challenging because the GNN model has many components in an architecture. So often from a paper, you cannot tell what are the setup in that architecture. So that will prohibit basically reproducing analysis in another study. So to address this goal, in a collaboration with our colleagues in Emory Computer Science Department, we have proposed a Brain GB. To the best of our knowledge, this is the first effort that tried to benchmark brain network analysis using GNMs. So essentially, it is a unified and modular reproducible framework for such kind of analysis that can help us facilitate reproducibility in the future studies. Basically, we can take FC network or structural network as input and feed them into the graph neural networks, GM, to perform downstream tasks, including predicting clinical outcomes or generate biomarkers. So the contribution of this benchmark work is, first of all, we summarize the pre-processing and construction pipelines for both type of networks, which will help to facilitate, the scientists to conduct the following analysis. And secondly, we take the architecture of the GNN and we decompose it into 4 manageable modules, and basically include a node feature selection, message passing, and attention mechanism and pooling methods, basically. For each of the module, we have considered different options by taking into account of the specific brain network structures. And then next, we have performed empirical studies using different data sets, and we have considered moderate style data versus large style data. And then we put together different pipelines by considering different choices in each of the modules in the GNM and compare the performances. From those results, we basically come up with some general recipes for how to design effective GNNs for brain network. For example, for the node feature selection module, we consider different type of ways to design the node features for brain network data, because for brain network data, all we have is connectivity between regions. So we don't have those node features as a direct input as in other network data. So how do you come up with the node features then for GNN, which is required in GNN? Basically, through our analysis, we find the connectivity profile is the best performing node feature, basically, for GNN. And similarly, we find out, the best performing options in other modules. So we have released this benchmark to the community. On this website, you can find the tutorials, and we also released this Python packages, and we released the pre-processing pipelines for SC and FC, and we also released trained models using the public available data. So basically, one of the goals of this benchmark is that after you standardize the mechanism for designing, GNN model or other type of deep learning model, it makes it much easier for people to replicate a specific analysis once you identify what are the options that is used in specific modules in the analysis pipeline. And then this will help you to build some foundation models or baseline models, which you can validate in other studies or further extends. So since it's a release of this work, it has attracted quite some attention in the community. So last time I checked, the paper has been cited over 160 times currently in the literature. So finally, just to talk about some challenges in deep learning in neuroimaging. So here are some commonly known of challenges. And the key limitation is a limited training data. So that is the biggest obstacle. And here are some methodology people have already developed to address this limitation. So I focus on specifically like a transfer learning kind of methods. So transfer learning has been successfully used in neuroimaging to try to address a limited training sample issue. So based on what we have seen, most of the transfer learning that's currently done are for structured neural image because they have a single image per subject, easier to handle. And the pre-trained model are derived from different sources. Some of the papers that use the pre-trained model that derived from non-imaging data sets like ImagingNet, and they also turned out to have some benefits and improve the performance to a certain extent. Other types of pre-training are done by using public available large-scale neuroimaging data sets. However, since there are only limited large-scale neuroimaging data sets, often the match is not exact. So some transfer learning are done by using cross-diagnosis type of transfer learning. Train a model using the ANI data, and then kind of a transfer learning it to study the Parkinson's disease. Some current challenges of transfer learning in neural images that most of the pre-trained model in the literature and in the field are based on 2D images. Transfer from 2D to 3D images is still pretty awkward, and this is mostly due to the lack of pre-trained 3D architecture and also due to a lack of appropriate method that can reliably transfer 2D knowledge to 3D images. So if we can address, the second limitation, I think it will help a lot. And finally, the transfer learning to fMRI is pretty rare because fMRI is actually beyond 3D, it's 4D. So this is still a pretty, underdeveloped method area. And many of the transfer learning involves collapse the fMRI into, a single image. Here are some softwares for connectivity and network analysis. And specifically, I want to highlight the first one, Toolbox, because this is the most commonly used connectivity analysis toolbox by neuroimaging community. The reason it's so popular is that it does everything. It takes the image, include the pre-processing and denoising in its pipeline, and it will extract different type of network metrics, and you conduct some, follow-up analysis. So one of the things I think this toolbox will be useful for statistical communities, that you can use this toolbox to help you generate some specific, connectivity measures, and then you can take those measures to conduct more sophisticated statistical modeling down the line. And some considerations, finally, some considerations, I think, in fMRI analysis. Develop advanced method is definitely a must, but at the same time, I want to emphasize the method also needs to be neurobiologically informed and motivated. It really should address unique characteristics of fMRI data and brain networks. That is when you would see real gain from a new method. Rigorous pre-processing and quality control is very crucial. It is a critical foundation for any downstream analysis because often you will see the effects from these upstream steps. They are so big, it actually overcome the effects you would see, differences you will see in when you choose different modeling mechanisms downstream. So and integrate, of course, fMRI with different modality. It is an important area. And finally, reliability and reproducibility needs to be significantly improved through some thorough validation. Like in every study, I think if possible, do some internal validation, like using data resampling, or external validation whenever possible, using a similar or comparable data set. See how your results stand up when you choose different tuning parameters. For example, in IC analysis, now we routinely kind of change the number of components that we extract from the data to study how robust the ICs were extracted under different parameter settings. And also for brain network analysis, how your study results will stand when you're using different atlas and connectivity measures is also a helpful thing to consider. I know it takes a lot of time, extra time after you're done with analysis. It seems like it's pretty challenging to spend extra time to consider this, but I think it's just to satisfy our own curiosity. It's worth to investigate it. And finally, I personally really think a validation section should be a built-in part of any imaging analysis papers, to increase the really the scientific relevance of the findings. So finally, I want to mention some resources and communities for people who want to get into neuroimaging analysis. And here are some major sources I want to mention. The American ASA Statistical Imaging section is a great community to get involved with. And here's our new website. In this community, we often have tutorials, and we have a monthly workshop, seminar, online webinar, and we have an annual conference. And basically, this is when you get involved with this community, it can significantly help statisticians to get started in imaging analysis. And then recently, a new resources, Stats AI, has been established. And this is a great resources and a community for imaging statisticians. So basically, in the website, they have provided a very comprehensive guidance to a different type of biomedical data, including neuroimaging data. So that is a great resource for people who want to work in this area. And finally, I want to acknowledge some acknowledgement. I'd like to thank the CBIS members, and especially my postdoc Yaotia Wang and my PhD student Guangming Yang, who have contributed to some contents in this talk. And Yaotia has a lot of experience in the pre-processing of and analysis of different imaging modalities. And he has generated some very insightful findings from our data. And I'd like to acknowledge my collaborators from the computer science department. Dr. Carl Yang, he is already a star in the field of GNN in computer science. And also her lab members, and they are the main drivers behind the BrainGB project. So I'd like to thank my research funding support from NIH and also Emory. That's all. Thank you.
Thank you, Ying. Really great talk and then really informative. It's already a little bit past 4, so
Sorry.
No. So I'm wondering if you have the room to take questions.
Oh, I'm fine. I'm fine. Yeah.
So great. So for the audience, so if you have any questions, so you can right now you can take the chance to ask Ying. You can either type in the chat or you can raise your hand and I will call your name. Yes. Aninda Roy, please unmute yourself and then ask question.
Hi. Hello, Dr. Wing. It is a really a great presentation, and I was like, because I started working on this fMRI data, and I was thinking one thing that I actually came to my mind, because I'm still learning, but it seems so when they actually work with the correlations, generate the correlations between two regions or in fMRI, like how actually they choose the threshold value of the correlations, like because they sometimes and I don't know like how actually it is statistically like how and another thing like when they actually but sometimes they use the adjacency matrix, which is um, trying to from the correlation they would use binary matrix, convert it to the binary matrix like 0 and 1 for the like to see the connections. Like I'm wondering like how threshold can affect if you want to map those um, structures or any metrics. Yes, please. Thanks.
Yeah. That's a great question. Yeah, basically, actually, I don't think I have a gold standard results or answer to that question because it's still open question in the field. So I think in neuroimaging communities, people hasn't really come up with the perfect method to thresholding. So some of the thresholding is based on the magnitude, like fMRI correlations, what kind of a magnitude is meaningful, and they want to capture. And other type of method is like they have used this kind of z-score type of method to do thresholding, that is to transform the whole connectivity matrix into a z, through the z transformation, conduct a z transformation. So you take the mean and standard deviation of all the connectivities in the matrix and then do a z transformation, then they threshold it based on a z-score. And for some other statistical methods, they have, for a Bayesian type of network modeling, for example, then people can use kind of credible, interval method to as a more like a systematic way or formal way to thresholding. And for partial correlation and those sparse controlled connecting matrix, this will be easy because after you do the sparsely control, that will help to already remove some of the many of the weak edges already. So that helps to clean up your connectivity matrices through that. So yeah, so it's basically, and in terms of generating from the, weighted connectivity matrix to the binary network matrix, similar answers, so it's based on specific quantitative methodologies and also based on some biological preferences the imaging scientist prefers. Yeah.
Yeah, just sorry. I know like there is like just for the following one just question is like So how important it is for like the people who are working actually in this area started working pretty recently. Uh, so how is getting the background of this study materials, like studies the brain networks and others, like how actually it can help, and um, is it real? Like, I think it is very important for. So, what do you think about it? Like, how much we need to know those study designs to analyze, and how it can be helpful to analyze the data?
Yes, that's a great question. I think it's actually very important to have some domain knowledge, in terms of the brain network, the different functional modules in the brain, for example. So because when you're looking at the brain atlas of oscillation, they have a 300 nodes. I give a simple example. So when you plotted that, make that a heat map of that brain connected matrix, how do you order in those nodes? That's already, you're right, it's not already straightforward. How do you order? Just order them randomly? Well, then you would have some very pretty, random looking, noisy looking, brain connectivity matrix that it doesn't offer any biological, insights. The ones that I show in my talk, you can see they typically have this kind of block diagonal pattern. I don't know whether you noticed. And that is because we order them based on the functional network modules. So basically, based on in the neuroimaging community, people already have identified some big brain networks of functional modules, in the brain. So you can group those nodes in your brain atlas into those functional modules. And then when you visualize your whole network, you show this kind of blocked type, heat map. And from that, there are some very interesting topological structures so that you can exploit actually in your statistical modeling. For example, in the statistical modeling, you can use a module information to come up with some module-specific models to reduce the parameters in your model. So you don't have to have, a huge number parameter for each edge, but rather you can have module-wise, model specifications, significantly reduce the parameters, but at the same time, you still captures the most relevant essential information. So, but those kind of insights in developing your model and your algorithm, that comes only have those neurological background. So I would highly advise, to not only read the papers in statistical community, but also in neuroscience, because they often offer some great insights. And you will build up your domain knowledge over the time. And personally, I actually think those domain knowledge are fascinating. That's what get me excited about neuroscience, to learn how different brain regions functions, it's kind of like, some really interesting facts. Yeah.
Thank you.
Yeah, and then Mingsha.
Oh yeah, can you hear me? This is Mixia Liu, from UNC Chapel Hill. Thank you very much for Dr. Guo. This is so nice talk. I really learned a lot. I have a question on the brain gene benchmark work. In this work will focus on the functional connectivity and the structural fusion using some message passing techniques. But in our experiment, especially in the for example, the early stage of the Alzheimer's, we found the structural connectivity may not change a lot, but the functional connectivity has changed. So their contribution may be different for the final prediction. So is there any thoughts for such kind of situation, and how can we balance these two modalities differently? Thank you.
Oh, yeah. That's a very great question. I don't think the current brain GB can do a lot of, cross-modality, multimodality fusion. So it's more like you have a specific modality and then you go through that pipeline for network construction, and then you can go through the, GNN. But I think there are other types of methods that people try to, combine the information from different modalities to, for prediction purposes. Actually, you're quite right. For different disease stages and also for different disease, different connectomes are important. For example, for Alzheimer, Parkinson type of neurological disease, we found like those structural imaging, structural connectomes, and has played a lot of if important role, but for those more like functionally related mental disorders, like psychiatry type of disorders, autism, schizophrenia, functional connectomes, there seems to have offers more predictive power. And also between functional MRI, I talk about task fMRI versus resting state fMRI. The resting state fMRI, the predictive power actually is not that very high for psychiatric disease. And if you can get a predictive R-square of 0.3, that's pretty common, actually. They already see that promising. But what people have found recently is that if you're using the right task that probe the specific disease mechanism, and then that task fMRI connectivity's prediction can have a better power than resting state fMRI, basically. So for example, if you are looking at schizophrenia, you use working memory type of connectivity. And when you're looking at a PTSD, you're looking at some kind of fear-related, task fMRI, you can have better predictive power. Yeah. I don't know whether that answers your question. Yeah.
Definitely. Thank you.
Sure.
Maybe one more question. So, Rajan? You are muted, so please unmute yourself.
Yeah, sorry.
Hi, Rajan.
Nice to see you. Hi. Hi. So I have a very sort of philosophical question, and it connects really to the first question here that was asked. So basically, everything is done, whether you do thresholding or you do these pre-assigned regions, and then you do the analysis, everything is done pretty much at a regional level, right?
Yeah.
And so is there a chance to move it to the voxel level, or is it such a horrendous problem that we don't even want to think about it?
Yeah. I think like a voxel level, of course, it will be like, the most has a highest spatial, right, resolutions. Yeah, and you have the most fine piece of information. The challenge of voxel level connectivity is that, as I mentioned, the fMRI data has some noises. So especially can you compare different subjects on the voxel level, the comparability between subjects are not that very high. So one voxel location in one subject may not corresponding very well to another subject's voxel, even though we normalize them to a template. So, and another challenge of that is that then your network will be huge because we can have hundreds of thousands of, so that will make the network a very challenging to deal with technically. And so that's why people is trying to coming up, with what is the best way, making sense way for us to group in those voxels. So that is what those functional atlas actually came into play. So basically, they're grouping those voxels based on how they're connected. Each voxel is connected with the rest of the brain. So each voxel, you have this vector of connectivity profile with the rest of all the other voxels in the brain, right? And then they would group the voxels based on those connectivity profiles of those voxels. So in a sense, voxels that have shared similar connectivity profile, they will be grouped together. So that has been found out to be a very effective way to best combine the voxels so that you don't lose too much spatial resolution because you're combining the most similar voxel. So you still get very consistent nodes, but at the same time, you significantly reduce the network size and help you to do computation. Yeah. So now the connectivity analysis, like almost all of them is atlas based. Yeah.
Okay, great. Thank you.
Sure.
Thank you. Very nice.
Thank you so much, Ying. Before I let you go, so just to make an announcement for our next seminar. So our next seminar will be on June 6th from 11 to 12 Eastern Time, offered by Professor Hernando Ombao. He's going to talk about another imaging modality, EEG data, which has like a higher temporal resolution but low spatial resolution, different from fMRI. And then please join us next time as well. And then thank you so much for attending this, and then see you next time. Thank you, Ying.
Thank you. Thank you, everyone, for your attention.
Bye.
Thank you very much. Great seminar. Thank you very much. Thank you.
Thank you.
Thank you.
Bye.
Thank you.